Okay, dear participants, good afternoon. It's my pleasure to act as a moderator to this webinar. My name is Teppo Laaksonen and I work as a network manager in Turku Science Park, city of Turku, southwest coast of Finland. Please note that this webinar will be recorded also. Later you will get access to the presentation shown in this webinar. First, I will give you a short introduction and background information for why we have this webinar going on. Yes, now we are a part of Biomarker Commercialization project. The project started 2017 and will be finalized by the end of this year. The project has been funded by European Regional Development Fund through Baltic Sea Interreg regional program. The budget for the project were roughly 2.5 million euros and the contribution from European Union 2 million euros roughly. In the project there were 8 different partner organizations from countries like Estonia, Finland, Germany, Lithuania, Poland, and Denmark. The coordinator for this project were from Denmark, Aalborg University Hospital. In this slide, you can see the logos of the 8 participating clusters. Like the name already tells, the aim for the project was to develop a set of tools for biomarker commercialization projects that are applicable for biomarkers. The tools are intended for use of researchers, companies, and of course technology transfer offices in universities. The tool it's a web-based tool and it's free of charge. The operational goal of this project were to develop and create 4 different type of tools. First one of them is commercialization guide, including also self-evaluation tool. And then best practices and pitfalls handbook. Third one, screening and selection guide coming soon, and then regulatory guide also. They can be found from internet, biomarker.nu. And like I told earlier, they are free of charge for users. Now this webinar is third webinar of the series of 3 different webinars. First one of those were held during June, how to commercialize your biomarker discovery. Second one were held during August, introduction to European IVD regulation. And now we are about to start the webinar number 3, launching the support tools for biomarker commercialization. And now I will give the screen to project leader Valerie Daussin from the University of Aalborg, Denmark. Please, Valerie.
Thank you, Teppo. My screen, yes. Can you see my screen now?
Yes, I can see.
Yes. Okay. Hello, everybody, and thank you for attending this third webinar. I hope not too many of you have already attended the first one because there will be not that many differences between that time and now. The most important difference is now we have a final tool in its final version, and that's what we will present to you. So thank you very much for your interest. I have prepare the presentation today in peace. So I will, of course, explain why we came with that project and what it was important for us as a group, as a consortium, to work on biomarker invention and their commercialization. And then I will make a demonstration of the big guide. And nevertheless, you got all a link, and I think Aalborg will send you a link again in the chat box. It just came now, and at some point we will just have a 5-10 minutes break, so you can have a look yourself, because I think it's a kind of tool you have just to have it in your hands if you really want to have an idea what it's about. Then I make a very short case story to illustrate a little bit the meaning with the tool and how it started, actually, because we had quite a few biomarkers invention in our portfolio at the hospital and wanted to find a better streamlining about how we were traveling, working further with them, developing and this kind of thing. And at the end, it will be more open for discussion and hear about your feedback and your question. And we will also see that what is the next step for us. So we decided to work on biomarker on the biological definition of them. When we started the project, we had a few discussions with our advisory board about, is that also digital biomarkers, which type? But then we decided it will be a too broad definition, and if we wanted to develop something useful, we had to concentrate on specific type of biomarkers. And in our case, and in agreement with all the partners, we decide to focus on the biological molecules related to biomarker inventions. So we had several of us, at least we were 3 TTOs in the group. We had some inventions in our portfolio based on those biomarkers inventions, and we were in that situation that some of them will be companion diagnostic, some will be more for making a specific diagnostic, or will be a predictive markers. There were very many applications, and they were a very broad type of markers you could be meeting in your everyday work. So when you are working in a TTO, and I think I will say that a little bit later also in the slide, you are dealing with a very large field. It can be anything, and the first question when you have an invention you need to evaluate, you have to ask, okay, what can it be used for? What is, at least for us as a hospital, what is a clinical need? So so that's a question who came every time, and we found it actually very difficult to have a structured process who were specific enough for the biomarkers, because they were a little bit different than other kind of medical device. I know they are not medical device, but compared to other process, they were mainly that category. But they were requiring a kind of other knowledge and deep questions. And then we had to build a development plan with a researcher, and we had to find the right development, how to prioritize. The funding is often very limited, so you need really to decide what are the most important next step if you want to transfer the technology later on. Most of the time we had also to decide how you will be transferring that in the clinical setting, how the laboratory will be able to use the test later on, and then what is the commercialization part of what you have done, because you may sitting with a lot of results that you need to sort out. If I need to explain a little bit about my own background, I have a legal background, work with APR all my experience. I have 20 years of experience, but I have worked with different area of technology, and the medical field was very new for me when I started at the hospital for 11 years ago. And then I found out that each invention was very unique in that field. And then when you sit like that with as a TTO and you get a biomarker inventions, yeah, sometimes it's a single marker, sometimes it will be a multiple marker. When it's a multiple marker, you have this question of ratio, and some of the time you have also a kind of artificial intelligence or algorithm to measure or to be able to pose a diagnostic after that. We had also this discrepancy between the technology platform, the markers were identified on and validated, and the technology platform you would have to transfer the results on for being able to use that on the in the clinic, because yeah, expensive equipment are not always available in the hospital. So if you want to have a commercial viable biomarker diagnostic at some point, you have to go into something which is, yeah, I said relatively cheap, but at least which is acceptable for the hospital to buy for. You have also to have this reliability, and the time can be also an issue. So you really need to think carefully with your team what will be the final product you would like to want to. When you work with invention generally, you have a lot of actors, a lot of people interested in the process. So of course you have the scientists, you have the technology transfer in most of the country who will be the first people to work with the scientists around the technology. But you then very quickly you will have the industrial partners, you will have eventually investors. So there is a lot of people to have to play together when you are working with scientists. Some are doctors, some are biotechnologists, and you have those people with very different kind of background that need to work together. The other reason, so we work with those biomarker commercialization, it's why is that we want to commercialize those inventions. And for us at the hospital, it was always been very obvious that if we want the patient to benefit of the research, there is no so many routes. It could be that you can develop an internal lab test in a hospital and use it for your own use. It begins to be more difficult if you have to make some tests from other hospital, or even more complicated if it's outside the country. But it seems that the new regulation will limit that quite a lot as well. So it may be an issue for many of the larger medical centers in the future to conduct their own measurement method. The regulation very complex. So when you want to go to the commercialization route, there is a lot of terms and concept and obligation that are very far from the experience we sit with in the hospital, either as scientists as a TTO. So it was also a very complex process when we were coming there. So at the end, we commercialize because we think that there is a clinical need and a value for the patient, and that's what we go after. At least that was our motivation to enter this project. So most of the time you sit there as a TTO and you need to get an idea, a general idea about the value of the invention you are trying to evaluate. You try to extrapolate already how you will be able to use it. It's not always easy to do that in early stage, but most of the time you can have an idea about your ideal situation where you want to use it. Is that a test bed? Is that in an ambulance you want to use it, or something like that? But it can be also a way also to understand those kind of pathway between the markers and the sickness, how they relate it to. What is most of the time difficult to bridge, and that's why us as TTO have an important role in the institution, is we need to help the researcher to present and convince others of the value how he has done. So you are really a facilitator and need to help them to provide the information in a way that the industry is interested to look into. So we found out actually, and that's what we found out when we started it, the project, that we discussed with many people. We discussed, of course, with a lot of scientists, but we discussed also with the industry and the investors, and trying to find out how was it typically that the collaboration were not successful, or why there were not more inventions in the diagnostic field reaching the market. But we found out that it's really difficult to have a common language. And even for me, who has been sitting now in a hospital for 11 years, I still use terms that some of my scientists are not really used to manipulate. So we really needed to have a kind of and on the other way, because I'm not a scientist, so I couldn't always be very sharp about communicating with them about the difficulties we had, for example, in losing some technical issue for going further in the development phase. So we wanted to have something which will support the understanding of each of the people involved in the process. The expertise are complementary, and that's, I think, an advantage is that everybody has its own expertise, because so there is no competition between the team. But it's really also important that we are good to play together, and that each one understand what it's expected from him and which is role in the larger team. So that's a little bit the background for developing the tool. We wanted to have a kind of visualization about the process, and you will see very soon that we try to look from the different perspective, and not necessarily from a user perspective, but from a commercial point of view. So the technical, the commercial, and the regulatory point of view. But also how we are interconnected, because most of the time as a TTO, you will never be able to work and develop and sell an invention unless the scientists are very an important part of the process. So we wanted to underline also that it's not necessarily the responsibility of one or the other, but it's a teamwork, and we need to complement each other very much for that. So we started to look at the process of the biomarkers inventions and how we bring them further in the process from the user perspective in the beginning. But we decide after around 1 year in the project, so that and with the help from our advisory board, that it will be more interesting to look from the technology point of view and look at the process that the technology needs to go from the invention to eventually a launch on the market. So I already say that, but really the idea was to create the overview and to start early and then try to guide the user through the process in milestones. We decided to follow the technology readiness levels in order to have also this kind of comparable items that the industry are usually using. So there were no existing proper TRL for the biomarker field. At least there were many versions of that, depending on which country we were into. So we had to develop a kind of TRL that were fitting with the kind of invention we were dealing with, and we wanted to have this regular and step-by-step process in order to limit the number of failure you have, because you haven't think about all the aspects. You have to go through the process, and it's a complex process. So it's easy to forget, for example, to apply for the right in your ethical committee approval to reuse, for example, some of the samples if you haven't thought about that. And it can be a common mistake sometimes if you are not really thinking a little bit further. So we wanted to make the user aware of those kind of elements in order to be better to anticipate what's next and what's next. So the big guide is supporting this process. And like I mentioned before, it's also because in a TTO you will not always have a molecular biology specialist. So you need to have a kind of support that can that can answer to several users who are not necessarily very high specialists in that specific field, because depending on the size of your TTO, you can't afford to have too many specialists. So and that's also a very specific area. So the big tool is, I will now tell what it is and what it is not, just to be sure about the expectation. It's a support tool. So and it's a support tool for a commercialization process. So I would say it's not a tool if you are a scientist who is not interested or doesn't see a clinical application for the results he's working on. Or if the results you are sitting with are not significant, that they will really support a commercialization process, because otherwise I think then the big guide is too heavy for just conducting your research like you will normally do it. But if you want to go into commercialization, then I think it's really really a very supportive tool to understand and to anticipate the next step. It doesn't tell you what to do or not to do. We had a lot of discussion about if we should have this kind of check, yes, I've done that, or if we should have an ordering about the task that the tool is guiding the user through. But it's not really what we wanted to. It's really, again, this idea about the overview, keeping track about where you are in the process and being able to anticipate. If you are going to apply for funding, it's really nice to know that, okay, within the next year or the next 2 years, that's where you want to go, or is that the goal you want to reach. So it's not about having very a very limited way of working the big guide. It's really a place to get inspiration and to get information, but it's not an internal process that you should implement at any cost if it's not fitting the way you are working. So it's really important to take part of the big guides which are relevant for you, and let over also some of the elements who are not. It can be also a large variation depending on the kind of invention you have. And I have experiences that myself when working with different kind of biomarker invention. Some of the tasks or some of the statistical calculation and everything can vary depending the context. So I think it's really important to use that as a support, but not as a must use. Otherwise, you can only be disappointed, I think. But it's keep you on the it's help you to keep a red line that you can follow through the process and avoid to be overwhelmed, because there is so many things to remember when you have to go further in the process. And I think it's really nice to support the communication you have always as a team, and also to find out at some point when do you need to invite in your group extra expertise if you are a little bit lacking in some of the aspects. In the big guide and also in the best practice handbook that I will present afterwards, we had also addressed the patenting issue, which are most of the time very complex. And biomarker has a little bit complex because there is at the moment still very difficulties with the US patenting of biomarkers, mainly because in the US definition, the biomarkers themselves are considered a product of nature. So it's really difficult, and you need to convert your patent formulation from a European version to a US version. So we wanted also to make that aware, trying to explain that in a little bit easy way. It will not replace the good advice you can get from a patent attorney, and it's really important to say that because it's a complex area. But we include there on the slide, if you go on the USPTO, they have those examples about the difference in formulation between different invention, what they can accept, what they cannot accept. But most of the time you will have to include the measurement method, the treatment steps, and those kind of things to convert your at least your patent eventually granted in Europe into something acceptable in the US. So that's also a part that you will find in the guide. Then I will go and show you the guide live. I think it's the best. Yes. Oh, I think yes, it's. Can you see all my my screen with the big guide? And I need to move a little bit. So you are on the page now for the big guide. So at the top, I don't know if I can show with a pen, maybe. No, I think I'm trying to see if I can show. Yeah, maybe. No. No, I will try to explain. At the top on the right, you will have very soon the website that we had before will be merged there, and then you will find general information about the big project and this kind of thing. But on this one, you will have the big guide, and you will find also the best practice handbook, the guide we developed for the regulation, the medical regulation on IVDR, and it will be available soon. Actually, I think it will be before the end of this week. And there is a link missing here. It will also appear in a few days. You will find also a review tool for making your own evaluation and finding what are the parameters that makes an invention a commercial viable invention. But if I click on the big guide, you arrive here and you have this process. So you have this discovery, verification, prototype, performance, who are the steps that we follow after the technology readiness level. I just need to say that at this stage, the two market phases have not been developed yet. The current big project is stopping at the end of this month. So actually we have a closing event on Friday, but we receive a further grant from the Interreg program, and we are starting in October for developing those two last phases that will be available next summer. We lacked a little bit competencies in our own consortium until now to be able to integrate what's going on when you are into the industry, because most of us as partner, we were more from the research area, but now we have included partners who have this knowledge and will be able to help us to build the two last phases. When you go on the right at the top, so you have an overview, and then you will open kind of scheme picture that show you all the process, and at least for the last first phases, and how how you move from one phase to the other, which kind of question you want to check, because if, for example, you don't have a clinical need very early you may have a difficulty to find a commercial value on your for example, on your invention. So this overview, it was just upload 10 minutes before we started, so it's new, but it's give you an overview, and it may be a nice tool to when you discuss and make some milestone checkup with your teams, how far you are. Everything is without having the necessity to go through the whole tool. I will try to check. Yeah. Then if I go on the first phase, for example, this discovery, like you said, it's answer to the technology readiness level 1 basic principle observed. And then we have developed the big tools, the big guide on 3 main track. The technical and clinical are one of them. And then we have some commercial steps. And at the end, we have the regulatory steps. We can say that for the first five, the regulatory is not a mandatory process, because it's mainly when you come with your final prototype that you need to go into your prototype that you want to have a clinical validation on, that the regulatory process will begin to be very mandatory. But we wanted to integrate the concept related to the regulatory much earlier, because sometimes they can really make the difference between a good collaboration with industry or not. So for example, if I click on that one, we had some consideration about ethical approval, because like I say, if you don't integrate an authorization for reusing the samples, and you can be there having those extra samples and you cannot use them because they were not validated from the beginning. So we tried to put some documents, for example, best practice on clinical samples. It's an extract from the best practice handbook, so everything is a little bit linked, but you will get some good advice that we got from the people related to a project. So most of them are people from the clinic who give them their good advice about how to deal with that. So all the tracks will follow those different kind of tasks. With an explanation about what we mean with the task. So for someone very well experienced in the research and working with biomarker, it may be very basic. But we discussed actually with quite a lot of the professor we had in our network, and they find it actually very useful. For example, if they had a student starting a PhD, having something to refer to in order to structure your project was actually very interesting for them. So we took it as a positive feedback, that even if you are very experienced, you can see the use of the tool for other people. So at the end of each task, so we have linked to relevant material. It could be, for example, the regulatory, the European information about that. It can be on document developed, for example, the best practice on patient consentment, and it can be also that you can get more information about something, for example, on another task, and then we have made some link. And you will find that for all the tasks, some have some extra material and some have not. Next to all those tasks, we have put those small icons that should be explained very soon about what they mean. But here, for example, yes, actually, I can see now, if I look at some point, yeah, I could see that it's related to an icon, because it's a scientific competence, you need that. And if you go there, it should be more business competence. And if you go, I think later on, but I need to go up there, it should be the regulatory competencies that you will have to add to your initial team. And the idea with that is really to under support that you can't solve the commercialization of such an invention by your own. So you really need to have a team around you that can complement the different competencies that will be necessary. And of course, under the scientific competencies, you can have many different kind of expertise. So it's only an indication, as well as under the business, you can have people with more business development background, but it can be also legal competencies. So just to remind that at some point you have to build a team around you, because that's what is the most important when you want to work with inventions. So that was for the tracks and how the tool is built. Then for each phases, we had make a small description about what is this technology readiness level, what we mean with that, how we have interpreted this kind of processing in our own process, and where we are at the end of each phase, is what is the achievement we have done when we are at the end of a phase. So just to, because it was not something which was streamlined already, because you didn't have the TRL for the biomarker field that much. So we wanted to be very clear about how we have interpreted all those concepts, just to be sure that everybody can speak the same language somewhere. Then we had a third place where we have put all the documents who are related to this specific phase. So in that case, it's the discovery phase. They will be under each of the tasks who are relevant, but we found it very nice to have a place where you could have this summary of the supporting document that are related to that. And at the end, we wanted to, before you move on with your project, sometimes it's really important just to make a small break and ask you again the same question. And it will be most of the time, sometimes it will be the same question, but it's because they are central for the commercialization success. If you don't have your clinical need, if you can't document it, so there is no commercial product. What is your market? What is the size? What is how will you position your technology in the existing clinical setup, the competition and everything. So those aspects, every time you reach a new phase in your technical development, you need to reassess also those aspects, because that's the one who are primarily related for the commercialization. So it's not enough to have good scientific results. You always need to check if the market is still relevant for the result you have achieved. So, and then the feasibility can be both technical or IPR or different aspects also there. So we have included this kind of self-evaluation, because we had that discussion as a group. You know, you will see also in a few minutes the self-evaluation tool, what we call the review tool. But for me, and in my experience, it's not always that easy to say we close a project or yes or no and answer that's black and white to a question, because there is so many parameters. You can end at the end of a phase and have not achieved what you expect, but maybe you can go back in the lab and do in a different way and that move forward. So the tool is not a black and white answer to do you have a commercial project. It's more, okay, if you reach these phases, so you increase your chance of success. And then of course you have the same principle and the same pattern in all the process until you come to the current fifth phase, which is at the moment the only one which is available. And some of the activity, of course, in the first phase, you have a very large technical and clinical track. When you come later in the process, so the commercial part or the business development part is going to filling. We had, for example, put some information about, okay, if you are going to license your technology, what are the typical terms that you will have to discuss with your partners. I know many countries we have a TTO that will support in that process, but we also know that even in Europe, it's not everywhere that the scientists have a TTO in their organization, or the role of a TTO can be different from a country to a country. So we wanted to give access to information for anybody, and independent who the user of the tool should be. Also because it's really important. Sometimes I have traveled with scientists many times in my career. When they sit here in a meeting room with people from the industry and they begin to discuss about all those terms, it's very nice when the scientists can feel at least involved and can follow up the discussion, because they can be very scared sometimes. It's really another world for them. So we wanted to provide this level of information so everybody can follow a part of the process, even if it's not your responsibility. At least you know why those discussions are occurring and what is the purpose of the next phase, for example. I will go back to the presentation. Yeah, I think it's more or less for now what I wanted to present about the big guide itself. And then I would like to propose 5 minutes. You got the link on the chat room for looking at the big guide yourself. If it's okay, just take those 5 minutes, look at it. Then I will illustrate a little bit the journey through a case, and then we will open up for discussion. 5 minutes break. Thank you. Okay, shall we start again? Tipu? Oh, yeah. Tipu, are you still here?
Yes, we can start. Yes.
Yeah.
I was unmuted.
That's fine. I was not, so I hope I didn't make too much noise.
No. Please continue.
There were a comment on the chat room that the tool is not available on small screen, and it's totally right. It was a choice we had to do with the developer because of the functionality. We were not able to keep the same functionality and quality if we were reducing the kind of screen you are going to look at the tool from. So you have to have at least, I think it's a certain 13-inch screen for being able to look at the tool. But I have myself a very small Lenovo, and it's working. But I know that if I'm on the smaller side, that I can't see it. You can't see it on an iPad, for example. It's too small. So yeah, that's a little bit the inconvenience, but we had to make some technical choice. So we were also thinking that most of the time when you are working in a lab, you often have access to a large screen. So we didn't see a special need to have a iPhone, a mobile phone or a tablet version. So maybe a future version, but right now it's a choice we did. I wanted to illustrate a little bit the big guide story with a case, because actually it's a very I think this invention I've been working on, it's probably the main reason why we entered the consortium as a university hospital. And some of you who are scientific or have a technical background maybe will find it a little bit strange on some aspect, but in my case, like I say, I have a legal background, so I have moved in many different kind of technology field, in the software, in the electronic, and so the last 10 years in the medical field. So every time it's a journey, because you have to open your knowledge to really new field in that level that you are able to understand. And the biomarkers invention actually have been very, very challenging for me. So at the beginning, I was thinking it's totally because I don't have the scientific background. But fortunately, discussing with other partners and other colleagues in Denmark and then building the consortium, I found out that it was not completely the only reason why it was difficult to work with biomarkers inventions. So for a few years ago, I've been in contact with this scientist who is now a professor. She was working for 15 years about the processing of the what we call the atherosclerosis, how the fat is depositing in your vein and in your bloodstream, building some plaques, and then at some point they detach and they form a clot, and then you have a kind of a lot of complication. We find out also that this protein CD36 was very linked to different kind of complication also in the organs, because of the deposit of the fat in the organs. So she found that the protein was available in a solid form in the plasma, and then she was able to measure the quantity of this protein, and being able to see the risk of certain patients of developing complication compared to other one. So that's mainly what she has been working on for a few years. And we had at some point 3 patents actually, with different methodology and application, how to measure the protein, but also how to associate that with different kind of complication. It was a very interesting and very well documented inventions at the time that she came to us. She had many publications. She had already actually patent from another institution, so we we took over the rights, but we were not with the technology at the time. She really preliminary invented, but we took over the right and tried to continue the commercialization process now that she was employed in our organization. And when she came to us, of course, it was a little bit more mature technology compared to other that we are working on, but it had everything to make it interesting as a commercialization process, because she has a very well developed method. She was very strong in her field. She had a lot of partnership across the world, actually measuring again this protein for other institutions and other research group, and being able to provide consistent and very regular results on that, and being associated with many publications that she was without any problem. She's a medical doctor, so she had no issues to explain what can be used, what are all the potential of this application, or of the application, and what was the potential for this protein. So in many ways it was a dream invention, because actually and it's a person who were very open for the commercialization process. She was very modest to say it's not really my field of expertise, but she was always willing to come up, to pitch, to visit the industry, and sometimes also to help me to identify who we should, and make a priority list about who we should discuss the first. The market potential was huge, and every time we were speaking with company at conferences, or in the beginning, and then at individual meetings, we were confirmed by the level of interest they had for this invention. What I didn't have at this time, I tried to open something, but maybe it's not working. I will open something else. What I didn't have at that time, maybe it is the one I'm trying to open now. Can you see a PDF file? Tepo, can you confirm if you see it?
Yes, I can see.
Yeah, the big review tool. Yeah.
Yes,
at that time, I didn't have a big review tool, and it was actually one of the challenging issues when you are not a specialist in the field, is how do you find the right level of questions? You don't want to begin to overplay your role, because you are not the scientist. You are not the scientist. They are the expert, but you have a sparring partner as a TTO. So it's always better if you have some qualified question in order to help them also to move on with their own process. I had some basic, of course, process to work on, and we had a lot of standard procedure and schema we were using for following the process. But I found really quickly that in the specific biomarker field, it was very limited. You had to be more specific, that if you are working with orthopedic prosthesis or something like that, it's a little bit more specific, because there is so many parameters, the medical applications, the biotechnology behind all this understanding of the pathophysiology and this kind of thing, which actually was not necessarily that easy to grasp when you are not really a specialist. So we had also one of the ambition we have for the big project it was also to develop, yes, a big guide, but also a kind of quick and dirty support tool for people like me who wants to always keep a track on the ultimate goal, which would be either to spin out or to transfer the technology to industry, or what it should be, but in securing also that you move forward and also bring the right question for the scientists, so they can themselves find out and identify what has a hurdle in the process. So we have developed this review tool where we decided together with our advisory board that at the end of the process, when you want to speak commercial, and you have three main questions you need always to answer: is there a clinical need? What is a commercial viability? And is that feasible? Is that something you will be able to measure in a practical routine? Are you accessible specimens? Is there different kind of conditions? So I can really say that I think at that time, if I had this review tool, I may have been a little bit sharper, and we may have avoided some issues in our process. I will go back to the presentation. So yeah, we had this technology, and we were very clear what we had and what we didn't have. But maybe we were a little bit naive about what we could expect from the other partners. So that we have this ELISA method, which was a little bit homemade because she was working with a pool of plasma, and it's not really what you prefer when you have to make a daily measurement. There was different aspect in her method which was not very you were not able to use that in a daily routine at the hospital when you have to do many tests in 1 hour. We think, on the contrary, that the method was simple enough that other research institutions will be able to use it by themselves. But we found out quickly that actually the partner we had, they prefer us to measure for them instead for trying to develop the method by themselves. So it was all of that was some of sign that we needed to something more standardized, more according to the industrial need. But we evaluate, and I think I still think that was right, that from our perspective we had enough material and enough documentation for moving forward. And what we thought the partner will be able to provide is maybe the competencies, the technical competencies that these inventors, who was a medical doctor, didn't have. It was all the understanding of the biochemistry process. She had this methodology, and she had some specific antibodies she had developed, and she had found commercially who were able to measure exactly the relationship between the atherosclerosis and the patient, and all this development of the condition. But we thought that it should be easy for an industrial partner who is used to develop antibodies and produce ELISA kit to transfer the technology. But we found actually quite quickly that it was not enough. Actually, we had very good relationship, and we had actually a very long-standing collaboration with a larger company in the diagnostic field. But actually it was much more complicated. I think we collaborated for almost 2 years, but at the end they were not able to transfer the technology. So just to make a story short, we are back in the lab trying now a different approach with alternative technology with another scientific group who are biotechnologists, and we will try to identify where on the proteins the antibodies are binding, because actually that's one of the things that we were not able to explain, and that makes the limitation for the industry to take over. But I think we were very clear in our process about what we wanted, but we were not really realistic. And I think it's mainly, and I really think that if I have been a little bit more sharper in understanding really what it's needed for transferring a biomarker invention into a commercialization process and the industry part of the process, maybe we will have make some choice earlier in the process instead for having able to do that later on in the process. So my my conclusion would be that, and of course you gain also a lot of experience by your mistakes, so it's also a good thing. But I think now the way we are working with invention, we are much more sharper to ask for the technical details that we were in the beginning. So that's a little bit at least the experience I wanted to share. So that's a little bit the story of the big guide. We, like I said, the project as it's in existing form now, it's stopping at the end of this month. But we got a grant for developing a second version, if I can say that. And the main goal for the next version is to finalize the development of the two last phase, who are more related to the industrial development of the IVD. We have had very interesting experience with the pilots, the people who have tested the big guide in its first version, that it will be interesting to adapt that with a project management tool. So that's also something we got the grant for. And in discussion with our advisory board, and some of them are working also in are from the university world. So of course they have a lot of training, educational responsibility. We found out that the big guide will be an interesting support also for training scientific into the commercialization process. So that's also the next phase of the development that we will have focus on, and the all should be achieved by June next year. At least I will just conclude that when I go back on the big guide, and I will go there now, at the top, so you have the big guide, and then you have this best practice handbook, which is available and that you can download here. It's maybe addressing a little bit more the TTOs, because we have tried really to be very honest about our success and pitfalls, like we call that in the guide, and try to have an explanation and a discussion with all the people we have involved in the collection of those best practice, about explaining why we had some failure and what we couldn't really be successful in our process. So not only making saying you should do that, but also, maybe if you don't do that, that's what the consequence will be. Because like I say, I think you learn that much from your failure, even if they are not very fun, but that's a part of life. And in the commercialization process, at least you can't have a success for every project you are working on, and there can be many different variables for success or not success, and each project is unique. So the teams, the people are unique, the technology maturity level is unique. In the scientific world, you often work with people who have vision, can see the future 10 years before the market is able. I've working with people working with supporting a process with algorithm and this kind of thing for more than 10 years, and the market, at least in the medical field, were not really ready for that for 10 years ago. It's completely another situation today. So you learn and you get better, but we wanted to share also all this knowledge with other people. And then by the end of the week, and now I have by the end of the week, we will have also the in vitro diagnostic regulatory guide, which try to explain the regulatory process in a scientific, accessible way. It can be very heavy to read the regulatory guidelines, so we wanted really to explain it and link it also to the process of maturing your invention. So it's taken in steps, following the seven steps of the big guide for that. Yes, that's what I could tell for today. I hope it was interesting enough, and
Yes.
we are open for discussion now.
Yeah, thank you very much, Valerie. This was very thorough and good presentation.
Thank you, Teppo.
Yeah, we don't. It was so thorough that we don't have any questions in the chat box. May I ask one question? You introduced that we will continue the project by 1 year until 2021. What happens then after that? Because clearly this is a huge amount of documentation that needs updating.
Yeah.
Every now and then.
Yeah, the extension is only for 9 months, but it's just a detail. But for us, it's not, because it's very short. So we will be finished by June. Actually, I hope we have developed the big guide in a way that it doesn't require too much updated, because none of the information is circumstantial, really. It's really like a book of cooking, you know. It's good. Yeah, it's general, and it takes a little more process. So I'm not sure it will get old that much. And yes, it could be some small links. Of course, we are linking to the, for example, the regulation in the IVD that we will have to keep track on. But actually, we are in a discussion with EATRIS Association for having them to take over somewhere the future of the big guide, because we have, of course, from the grant from the Interreg program, we will keep the website and everything running for at least 5 years. I think if it's the obligation, at least we have. And I will also say that we have an enormous interest ourselves as user, as a university hospital. So we are completely motivated to keep the big guide running, but we want to secure also its future and also the access for the right population. So we really hope that we will find the right way for introducing it on the existing platform at the European level for the scientific working in the biomarker area.
Okay, thank you very much. I think now it's time to continue. We are a little bit over time, so this is good time to conclude and stop this webinar. Thank you very much for all participation, and specifically to Valerie, you. Thank you.
Thank you, Teppo. Bye-bye.
Bye-bye.
Goodbye.